bachelor degrees in biochemistry and genetics. Did research throughout undergrad in the lab of Andrea Elmafatano, and I apologize if I butchered that. 3 publications from his work, one co-first author, including one that was co-first author. Joined BCM as a member of the MD PhD program in 2020. And after a 2-year hiatus from research due to med school and pandemic, he joined Will's lab in his first year of graduate school, where now he works on mouse models and human tumor samples to study T-cell exhaustion. I'm interested in oncology and pathology. And with that, I'm going to give it over to Will to give his part of the talk.
Thank you, Joel. Can you all hear me okay? Okay, great. All right, so thanks. As Joel said, I'm an assistant professor across the road at Baylor College of Medicine. Since I'm going first, I'll just put acknowledgement slides here at the beginning. I'm really thankful for our funding sources and our lab team. There are 6 of us, and Sean is a grad student in the lab who will be talking next. We're very interested in T-cell exhaustion. I think you had a really nice introduction from Mauro earlier about what T-cell exhaustion is. It is the process by which, in particular, we're interested in CD8 T cells become dysfunctional over time, and that prevents the clearance of chronic viral infections and cancer. And reversing T-cell exhaustion or overcoming T-cell exhaustion is a very important treatment modality for cancer in particular. So I'll just give one slide of background about what CD8 T cells in cancer do. All of you probably know all this, but CD8 T cells recognize antigen through MHC1. A tumor cell or an infected cell will present a foreign peptide on MHC. The T cell receptor of that CD8 T cell will recognize it and then ideally secrete perforin, granzymes, different cytokines to kill that cell. But of course, tumor growth is promoted by pathways that inhibit this process and cause T cell exhaustion. And as Mario also pointed out, there's a considerable diversity in antigen specificity and also differentiation states within the tumor microenvironment, especially in humans. You don't always know what you're going to get inside the tumor microenvironment. So at my previous institution, we were really interested in CD8 T cell responses in humans and in particular in brain metastases. So about 20% of patients with cancer will develop brain metastases. That number could be higher, but they're just not found because of lack of screening. This is obviously a cause of significant morbidity, mortality. But the good news is many of these are actually responsive to immune checkpoint inhibition, especially with PD-1 pathway blockade and CTLA-4. So at Emory University, we had access to, or we actually received freshly resected tumor samples from 31 patients with brain metastases who were immunotherapy naive. And our goal was to identify immunotherapy responsive T cell populations in these tumors and identify what markers might be expressed on them and potential pathways that could be targeted for brain metastasis-specific immunotherapy. So I'll talk a little bit about what we did. Again, we had a patient cohort of 31 patients. We received these tumors right as they came out of the patient, so the surgeons were great. They dropped it in the media and handed it right to us, so we got these samples very fresh. We also got paired blood from these patients at the time of surgical resection. So from the tumors themselves, we froze part of the tissue for spatial transcriptomics, which I'm not going to talk about. But from the rest of it, we digested it with a cocktail of enzymes and generated a single cell suspension. And on that, I will talk about today, we did flow cytometry as well as single cell RNA sequencing. We also did a very small flow cytometry panel directly out of the patient so we could quantify the number of T cells and other immune cells that were in the patient. So we did that without washing the flow. The staining is not the best, but you actually get an exact number of how many T cells or other cells were in that patient. And then we also did flow and TCR sequencing from the blood. And what we found is that, brain metastases are actually fairly well infiltrated by CD8 T cells in humans, although this does vary based on the primary tumor type. And so I'm showing here, this is the number of just lymphocytes per gram of tumor, and over here on the right, the number of CD8 T cells per gram of tumor. You see there's a very wide spread, but a lot of this is actually explained by the etiology of the primary tumor. So breast tumors were quite cold, which mimics the primary tumors in this patient population. And melanoma was actually got a very large number of CD8 T cells out of the patients with these brain metastases. So we had some questions that we wanted to answer in this study, and this is published if you want to go back and look. But the main one I'll talk about here are kind of the first and the fourth. are what is the phenotype of brain metastasis infiltrating CD8 T cells? Can we find those progenitor cells that Mara talked about? What markers are these expressing, and could it be amenable to checkpoint blockade or other therapies? And then what are the specific T cell clones that are located within the TME? So we were working on this panel about 4 years ago, and we had access to a 4-laser Cytek Aurora. We didn't have the UV. We have access to a 5-laser instrument now, so we're trying to build this panel out a little bit. But we were very interested in a CD8 T cell centric panel to look at CD8 T cell state within these tissues. So if you look here on the right, you won't even see CD19. We don't really have any any markers of B cells or other myeloid cells, as we're mainly interested in CD8 T cells. It took us 8 iterations to get optimal pairing. So that's just essentially just moving colors around. Of course, we were titrating and things like that. And you'll probably see here, if you look at this, there is space where you could put in something spectrally, but almost all of these markers are co-expressed. Almost all of these are on T cells, especially on exhausted T cells. The TILs, that is the tumor infiltrating lymphocytes that we got from these patients, those are very precious. You can't get more of it. And so we would optimize this panel on PBMCs that you could get from healthy donors. That can be problematic because some of these proteins like TIM3 aren't expressed at high levels on CD8 T cells or other cell populations in the blood. And so you have to make sure that you actually go back to TILs to make sure these antibodies work, because you may get negative staining just because that marker isn't present in the blood. And I think you've had some great talks both earlier today and yesterday about how to design these panels, so I'm talking about that. One thing that we found is that we could sort of use secondary reagents to get some creative color combinations. For example, we used a biotinylated granzyme B antibody, and then you can use a streptavidin in any color you want, essentially, to measure granzyme B. The TCF1 antibody here is a rabbit antibody, and it's the only one in this panel, so you can use an anti-rabbit secondary, and you can get that in many different colors. We weren't that creative, but those secondary reagents really helped us fit in more things into our panel, in some cases made the panel look better. So I'm going to show you some of the plots. I'm going to get into dimensionality reduction and things like that, but I really like to go through and look at the individual marker expressions, because when you know about the biology of the T cell or the cell that you're focused on, you know which cell should stain for what, and it kind of gives you an idea, not only of the biology, but how well your panel is working. So this is, these are our blood. This is PBMCs from the matched patient in which we analyze tumor-infiltrating T cells over here on the right and the bottom. So you have this very nice population of naive CD8 T cells, which are double positive for CCR7 and CD44RA, but you don't really have these naive cells in the tumor, which is consistent with earlier data. And so it shows you that you're already getting selective infiltration of brain metastases by non-naive CD8 T cells. And I just like to look at everything versus PD-1, because PD-1 is essentially a marker of T cell activation, but of course it's very high on exhausted T cells as well. And so you can see that generally the PD-1 positive cells were negative for CD45RA. There were some that expressed TCF1, which is indicative that they could be stim-like. Some for CD127, and then of course they express a lot of these checkpoint molecules like CTLA4, TIM3, but they also express some effector molecules such as granzyme B. They were positive and negative for the costimulatory molecule, CD28. But of course, it's very hard to look at N-by-N plots, as mentioned earlier, when you have this many colors. And so we did UMAP dimensionality reduction on this. And so the way that we collected these samples is that we freeze them, right? So we get blood from these patients, we isolate the TILs, we viably cryopreserve them, we thaw them all at once so that we can run flow all at once without any sort of batch effect. Even then, I've discovered, flow is much harder on human samples, especially on tumor infiltrating samples, than it is on mice, just because there's a lot of heterogeneity between patients, but also how the surgery went, how long did it take for that sample to come from pathology, things like that. But we found 6 broad clusters of CD8 T cells in these patients, looking at the blood and the lung. And what I'm showing here is 4 different primary tumors. Again, these are all brain metastases, but 4 different primary etiologies. And looking at the TILs and also looking at you see that the T cells are very different. So I'll show you in a second that these gray cells are naive. You see a lot of them in the blood of these patients, but you don't really see a lot in the tumor. So what are these clusters actually? Well, as I mentioned earlier, cluster 1, you can see that it's negative for PD-1, negative for CD39. It has a lot of CD127, which is IL-7 receptor, and of course, it's the only one expressing CCR7, so these are naive. The other easy one to identify are these orange ones. You can see they're very high for Ki67, which is down here. They also express HLA-DR, which is MHC class 2, so they're very activated, but they are also expressing things like CD39 and PD-1. The highest cells for PD-1 are this red population, and you can see that these are very classically exhausted. They have a little bit of granzyme B, but they have high levels of tox, TIM3, CTLA4. and so on. So we were interested which of these populations infiltrated different tissues. And so what I'm showing here, essentially, this is the statistics here were done in a paired manner between patient blood and tumor. I didn't show the lines because it gets really busy, but the closed circle here is blood, the open circle is tumor. And you can see that you have a lot of naive cells in the blood, average around like 30% or so, and then pretty much none of those get into the tumor. And then you have two populations that are very high in the tumor. That's this cluster 4, and this looks like some sort of tissue resident memory T cell. You see that they do express some PD-1, but they're lower than the exhausted T cells. And then you have cluster 5 here, which is that exhausted population, which is very bright for PD. And so, like the previous couple talks, we also wanted to go back and do what we consider really a complementary approach, which is single-cell RNA sequencing. And we focus on PD-1 positive CD8 T cells because these are the ones that are most likely to be antigen specific, although it's important to note that not all of these are antigen specific, and we could show that experimentally. But we actually saw really similar clusters, right? So we kind of spiked in some naive cells from the PBMCs of these patients as a control. This clustered together nicely. They had very high levels of CCR7, for example. And then we found 4 clusters, which was called A, B, C, and dividing. And it's very similar to what we saw by the flow, right? You have this one population that has high Ki67. It's also expressing high levels of exhaustion markers like CTLA4 and LAG3. And then you have these other 2 populations, B and C, which are kind of similar. They look more like a memory population, and they're expressing a lot of IL-7 receptor. And so what this can't tell you and what flow can't tell you, but what we're really interested in is how are these T cell subsets related? Are these cells in B and C differentiating into cluster A, or are these separate populations that just don't see the same antigen? And how we can do this is sequence the T cell receptor. If you recall from that earlier slide, an individual T cell receptor is what dictates the antigen specificity of that T cell, what it can actually see. And so if 2 cells have the same T cell receptor, they can differentiate from one state to the other. But if 2 cells don't have the same T cell receptor, that means they're seeing completely different antigens. And we can measure the overlap between T cell populations and their T cell receptors with this index called the Morishita-Forin index. So 1 means you have completely identical TCRs between populations, and 0 means you have absolutely no overlap. And so if you look at here, you have these exhausted cells and you have these less exhausted cells. There's almost no overlap between the exhausted cells and the non-exhausted cells. And so what that means is they're seeing completely different populations. They're not differentiating from one state to the other. And so the second half of this relatively short human talk, I want to talk about some of the interesting things that you can get as you prospectively collect these samples. So again, we're just getting them from the surgeons and say, hey, we've got a sample today, and we process it and put it in the freezer. But we ended up getting 2 samples from the same patient. This is a very unusual case. This doesn't happen very often. This was a patient with actually 4 non-small cell lung cancer brain metastases. There was one particularly large tumor. It was resected without any prior treatment, and the resection cavity and 3 other metastases were irradiated. One of these progressed through 30 gray of radiation, and then about a week after the cessation of radiation, that was actually resected as well. And so we have blood at both times, and we have a tumor from the same patient that has been irradiated and one that has not. And so this is like an immunologist's dream to have from a human patient, because you obviously don't do craniotomies to open up people's skulls very often. This is what it looks like. This was the first tumor that was resected. You can see that it goes away and stays gone after treatment. This is right before surgery. This is the second lesion, and you can see that it continues to get bigger through radiation, and it was eventually resected as well. And this is the treatment field showing that it was actually irradiated. And so we did our conventional flow cytometry, and that showed that we had reduced immune infiltrate in the irradiated lesion. Total lymphocytes were decreased by about 7-fold, and it was about 17 times lower CD8 T cells in that irradiated tumor, which indicates a significant depletion of the CD8 T cell population. I don't have time to show the data, but the TCRs were completely different, meaning that all the T cells that were there were killed by radiation and new ones came in. But I want to talk a little bit about the CytSeq data that we did. And so many of you may know this is using oligonucleotide labeled antibodies. Again, we kind of consider this complementary to flow cytometry because it's very expensive. You can't do many cells, but you can get a very large number of parameters without compensation. And so we used BioLegend's TotalSeq cocktail, which is 130 different antigens. So you can measure protein expression and RNA expression and T cell receptors on single cells. And so we sorted CD45 positive antibodies, and we did have to be careful. You don't want to use the same CD45 antibody that's in your universal cocktail kit. And so Milton actually has this really nice resource called extended validation. And so there was this clone here that did not cross-react with HI30, which is a common CD45 clone. So we use HI30 to sort out these T cells, these immune cells, these CD45 positive cells, and then use this one to sort. And so just using the site-seq data, this is only protein data here. This is 13 sort of broad clusters that are present in the blood and the tumor. We sequence both. And you can see you have a very large population of monocytes. These are primarily in the blood. You have macrophages. These are almost all in the tumor. Some B cells, and then I've colored blue here, CD8 T cells, and then green, CD4 T cells. And so what we saw, without getting too much into the details, is that you did have quite large differences, as you might expect, between the untreated tumor and the unirradiated tumor. So in particular, you had this dark blue subset, which down here, these are actually exhausted CD8 T cells. These are very high for PD-1. They're very high for CD39. They're very clonal. So they're obviously seeing some sort of antigen in the tumor. These were completely gone between the before and after radiation. And what you got instead is this very large infiltrate of macrophages. And you can actually see that they cluster together. This dark red is from the irradiated tumor. This dark blue is from the untreated tumor. And it looks like monocytes started to actually migrate into this tumor. They were very high for IL-1. And so they started to actually migrate into the tumor and start to differentiate to macrophages. So there were lots of interesting changes here, but I kind of wanted to show you a little bit more about the technique. And so you can gate this just like conventional flow cytometry, right? So you can choose CD4 and CD19. I think I'll show you that in a second. But I was really impressed by the ability to detect rare T cell subsets. So these are two different TCRs that indicate mate cells and also a class of gamma delta T cells. And this is looking at all cells from this whole sample. So there are like 60,000 cells here. And you can very clearly see what these clusters are or what these populations are. And they're colored by the cluster. So if you remember, CD8s are in blue. So most of your gamma delta T cells, or this particular subset of gamma delta T cells in the tumor, are CD8s, and then blue are CD4. So you're getting both CD8 and CD4 positive mates in these tumors. And so this was done in R, not in FlowJo. I don't know if it's possible in FlowJo, but it might be. And like I said, you can conventionally gate this just like anything else. So this is looking at all cells. This is CD19 versus CD3. I don't know where my label went. So these are T cells. You can gate on that, and you see CD4 versus CD8. And you see there's no, there are some double positive cells, but you don't have any compensation to worry about. This is all sequencing. It's all independent of each other. And again, looking at the CD8 T cells, as I mentioned, those exhausted cells went away after irradiation. And so what I'm showing here is each dot is a cell. Blue is from the unirradiated tumor, the untreated tumor. Red is from the treated tumor. And you see that you have this very large population of CD39, PD-1 high T cells in the untreated tumor. And then in the irradiated tumor, you have all PD-1 negative, CD39 negative, largely CD69 negative cells, which means there's just cells from the circulation that are coming into this tumor after the fact. And we were able to back that up by TCR sequencing, which showed that these red TILs actually shared TCR clones with the circulating T cells. So before I get over to Sean here, just a couple of things about here. I think prospectively collecting these tumors can lead to very interesting things. Irradiation, dramatically affected the T cell populations that we saw in these tumors, and particularly the likely loss of tumor-specific CD8 T cells. And so our goals with all these studies are to, gain a better understanding of human anti-tumor T cell responses. But of course, we're limited in what we can do with human samples. And so we use a lot of in vivo mouse models of exhaustion as well. And our goal is to identify new therapeutic strategies that can improve T cell function in chronic viral infection cancer. And so a lot of the things that we observe in these models, we take back to the mouse models. And that's what Sean's going to talk about. And he's going to talk about CD7, which is this interesting molecule that's actually expressed on most CD8 T cells. But we see the highest expression on exhausted CD8 T cells. And we saw that in our mouse models of infection too. And so his project is really to look at the role of CD7 in T cell exhaustion. And I'll let him take it away if I can switch the PowerPoint. There we go. You may. Yeah, so
Our next speaker is Matilda Mostrom. um, Matilda completed her bachelor's degree in biology at the University of Louisiana at Lafayette in 2017, and a PhD in biomedical sciences at Tulane University in 2022. And her background is in immunology with specific focus on the maternal-fetal interface and immune responses to viral infection in pregnancy. She has high parameter panel design experience by developing an optimized multicolor immunofluorescence panel, the OMIP, which is the standard in the field and then the first broad immunophenotyping in non-human primates. As an assistant director of the flow cytometry core at the Tulane National Primate Research Center, she's bringing up appropriate standards to apply for the International Society for Advancement of Cytometry Shared Resource Laboratory Recognition Program. And as we all are familiar with the SRL recognition program. And Matilda is currently participating in the NPRC consortium non-human primate flow collaboration project with Beckman Dickinson, the BD, to characterize the frequency of over 100 targets in major immune cell subsets of non-human primate species. In addition, Matilda is reinventing the user training at Tulane National Primate Research Center to increase data quality, while also improving the throughput of users in the training program for self-acquisition. Welcome, Matilda.
All right. Thank you, everyone, for inviting me to give this talk. To the organizers, this is a great conference. I'm really excited to be here. And I know we're on day 3, so we're getting tired. So I just want to bring some energy back to the room by doing a show of hands. How many people here know about congenital CMV? Sure. Yeah, very good, very good. And of those people who are aware of congenital CMV, do you believe that congenital CMV should be screened for in neonatal screening? Yeah, that'd be a good thing. All right. And so for you who are here and are not so aware of congenital CMV, you're not alone. This is a survey in the US of women where they're aware of a certain disease in newborns versus the incidence rate. And you can compare congenital CMV with that of Down syndrome in terms of incidence, but the awareness is quite low. What happens when there is congenital CMV? I'll mention that congenital is a word used to describe something that occurs during pregnancy. So a congenital infection is an infection that occurs from mother to fetus during pregnancy. And in newborns, they can either be asymptomatic or be unfortunately symptomatic for the disease. Some of this is resolved, some are permanent outcomes, and this can vary from fetal death to hearing loss. Hearing loss is the most common. We currently don't know why this happens in some women and not in other women. We have no well-known correlates of protection and no vaccines against this. But as many of us who study immunology know that the circulation rarely reflects the state of the tissue. And I would argue that the maternal-fetal interface is very much the case in terms of tissue, even maybe more so than other tissues, because we need to have a support for the fetal growth to tolerate any of the paternal antigens in the placenta. And from any infections in the mother, there needs to be a protection here to prevent the fetus from getting infected. And in humans, this is quite hard to sample because, as I mentioned, CMV is not screened for. And so getting samples from humans to study this is quite hard. And this is when an animal model could be really powerful to help us gain preclinical correlates of protection. And we can, in this animal model in non-human primates, of rhesus macaques, we have a closely genetically similar host and virus and similar placental development. So at Tulane National Primate Research Center, we've established a model to do this. So we are posed to ask questions like, what immune cells are required for control of the virus and protecting subsequently the fetus from congenital CMV? So we've published on this in the past in a CD4 T cell depletion model. And I'll emphasize that in our macaques, the animals are naturally infected through co-housing in the colony. Just like in humans, CMV is readily spread through shedding of body fluids that other animals are exposed to at a young age and before childbearing age, many times in humans, or sexual maturity in the animals, they've already been acquired the virus, and we then deplete these animals. No experimental infection. So this is our data. Here on the left, you're looking at the depletion kinetics for absolute counts for CD4 T cells. These animals are in the first trimester of pregnancy. We do a one-time infusion of anti-CD4, and this leads to a nice depletion in the peripheral blood. On the right, you're looking at the viral loads in plasma and amniotic fluid. No virus detected. The animals are readily shedding virus in saliva and urine, however. But these animals are controlling the virus, and the bottom here is the gold standard for diagnosing congenital CMV. We have not detected this. So we want to move on now in new studies. What role does the cytotoxic immune system play? So I'll reiterate, we don't have immune correlates protection for this disease. But we do know, however, that CMV encodes several immune evasion genes for NK and CD8 T cell responses. And those same cells are enriched at the maternal-fetal interface. So what is their role in protection? Are they required for protecting the fetus against congenital CMV? So to address this, we have done in vivo depletions of NK and CD8 T cells in pregnant seropositive rhesus macaques. Now, how do you deplete NK cells in rhesus macaques? There's a cytokine called IL-15. The IL-15 receptor is expressed on NK cells, and it's essential for both development and survival of NK cells. But also, it plays an important role in maintenance of T cell effector memory. So that's something to take a look at. This is the study design. We have 4 CMV positive dams in their first trimester of pregnancy. They received 4 doses of anti-IL-15. And the first animal we performed this in, she had a fetal death at day 7. I'm going to come back and talk to this. The other animals went on to study planned C-section. Let's first start with the data from the depletion itself. On the left, you're looking at our collaborators' data where we shipped samples to them. They performed the NK cell phenotyping. And here you're looking at the pre-gating up to the final gate here with NKG2A staining. In non-human primates, the NKG2A binding antibody cross-reacts with NKG2C. So it's really a combination of the 2 markers. And this is the way to phenotype NK cells. We see in the PBMCs that we have a nice depletion that's maintained through this dosing. And in tissue biopsies like colorectal and peripheral lymph node, we have a nice depletion as well. It's not 100%, but I'd say it's a pretty vast depletion, the NK cells. Now, I promised you effector memory. So this is data on the left of CD4 effector memory and on the right, CD8 effector memory. And there's a lot of animal-to-animal variation here, but I would describe that we're observing a decrease in the frequency of CD8 effector memory. That's transient, and there's a lot of animal-to-animal variation in terms of how long it went on and the extent. But what happened to the control of the virus? On the left, you're looking at the peripheral viral load. In these animals, we see that 3 out of 4 have peripheral viral load. This is reactivation of virus. This is the first time we've seen this in a depletion alone model. On the right, you're looking at that gold standard diagnostic. None of our animals come back with positive PCR for amniotic fluid. We dug further and looked into the tissues. On the left, you're looking at the first animal that had that spontaneous abortion. And the veterinarians were able to collect as much tissue as possible as she was shedding placenta. And we performed qPCR on all those tissue pieces. They were all negative for CMV. On the right were the animals that went to term, where we look in fetal tissues, including the placenta and the maternal decidua. They were all negative. So now let's move to our second cohort. How do you deplete CD8 T cells in rhesus macaques? And And I want to pause here and talk about CD8 for a bit because this molecule can be expressed as either a heterodimer or a homodimer, alpha beta chain or alpha alpha chain together. Your traditional CD8 T cell that you think of as a CD8 T cell are CD8 heterodimer expressors, while most NK cells are homodimer expression. So when you look in a journal and you look at, oh, there's a CD8 T cell depletion, you want to pay attention to what chain the depletion treatment is targeting. We chose to use anti-CD8 beta. This is our study design. In 3 animals, we've infused them with anti-CD8 beta. And when you stain for this to monitor the depletion outcome, the most common non-human primate clone for CD8 is the SK1 clone. That's only binding CD8 alpha epitope. But the BD and Beckman Coulter clones shown here, long names, they bind a epitope that's only found when CD8 exists as a heterodimer. So that's what you want to use. This is the gating strategy from our lab for a time, single, forward size scatter. Then whether or not the cells express gamma delta, these are assumed to be your gamma delta T cells, and the negative to be your alpha beta T cells, traditional. On the top here, you're looking at the co-stain of the heterodimer or the alpha staining antibody alone. So you're double positive, those are our heterodimer. Majority of alpha beta is heterodimer. Gamma delta T cells, this is similar to humans, are mostly homodimer. The NK cells gated on the bottom really don't see much heterodimer. This is the absolute T cell counts for that heterodimer, and we see a nice depletion over time, where we have one animal here kind of regaining some of that population by 100 days post-depletion. These are the other CD8 T cell expressing subsets of the alpha-alpha homodimer, gamma delta, and NK cells. Unfortunately, I don't have yet data gated out or the analysis for absolute counts here, so bear with me. But the point I want to make here is that the other CD8 expressing cells are not disappearing, but it's a question of, are they actually expanding? We don't know because this is not absolute counts. This is percentages, which would be impacted by removing another subset. So to be continued. How did the control of the virus go? Left plasma, we see that no animals have any reactivation. We have one positive time point here at day 0 in one animal, which we're investigating further if it's sample mix-up, contamination, or a real animal stress from changing housing that led to this. No amniotic fluid came back positive. But when we look in the tissue in this cohort, we see for the first time that LC89 here in black has positive fetal urinary bladder, positive amniotic membrane of the placenta. The primary and secondary placenta are positive, as well as that last layer from the mom, the maternal decidua. So what happened at the maternal fetal interface? This is gating at the decidua. So I'll come back here and mention that the decidua is in pregnancy when the endometrium undergoes decidulation. As the embryo invades the uterine wall and develops the placenta, that's the maternal tissue of the placenta, which is rich in immune cells. So we have time, 45, forward size scatter, live, dead, single. Then with CD3 alone, I didn't have gamma delta in this panel. We see that there's really a lack of heterodimer, and same for the NK cells. And I'll point out here that on the top is the same data from the gating strategy. In the fetal tissues, here in fetal inguinal lymph node, we can detect heterodimer. So it appears to be that there's a long-term depletion in the decidua. But the question is, what is normal? Because we don't have that data in a cohesive way. So what's next is that I'm redesigning my omic for a specific large batch experiment across all 3 cohorts of true untreated controls, the 2 cohorts I mentioned here. What is the NK and CD8 heterodimer frequencies? So with that, I'll conclude that the anti-IL-15 treatment led to profound depletion. There was a short-lived reduction of CD8 effector memory, and we did see reactivation in this cohort. CD8 beta treatment, however, led to no viremia that we could detect despite that nice depletion. The other CD8 expressing cells were maintained. And And we have one case of congenital CMV, which is suggestive of a lack of tissue level control at the maternal fetal interface. And this is all in contrast to our earlier work where CD4 T cell depletion led to neither viremia or congenital CMV. So with that, I'll thank the Macaque lab where I did my PhD and continuing this work with, and the flow cytometry core where I now work, past and current staff I'd like to thank, and our collaborators at Cornell and Duke in the lab of Sally Permar and Keith Reeves, and of course the veterinary staff and the animals that make this work possible. So with that, I'll take any questions. Yeah, I'm going to repeat the question. So the question from Veronica is, what type of CD8 T cell is important? And that's a very interesting question. I have, from my work, looking at normal, what is normal? The maternal fetal interface is really rich in effector memory and really more inflammatory type T cells. And so by removing our CD heterodimer, I think those, which are high frequency there, are being lost. And that might be why we see that transmission. But I'm not sure until we do further investigation. Thank you.
Yeah, I was wondering, very interesting work to be able to do those. Concerns that I would have responses are that if you wipe out major proportions of the lymphocytes, you'll end up with homeostatic proliferation, which can definitely do things like increase CD4 expression, and what that does functionally is,
Right.
I don't personally know that. Maybe you or speak to it.
Mm-hmm.
Functional blockade rather than
Mm. Yeah, no. I agree. So if I summarize the question, I don't know if my microphone's working. Are we, by removing cells, having a proliferative response that's changing the immune system? And then the second part was kind of, could we do a different type of depletion? So to the first part, I'll mention that, yeah, there's no perfect depletion model. It's like removing a huge, and IL-15, for example, is expressed, the receptor is expressed on other cells. So yes, for sure, we could be altering the immune system in a different way. We are attempting to do more like antiviral functional responses, but the problem we end up with is that sampling these animals during pregnancy, we don't get that much blood, and then from freezing, we have very little cells back. So yeah, we've done some ELISpots, and we see in the IL-15 group, we do see reduction in the anti-CMV specific immune responses. They're not completely gone, but they are reduced, so that might play a role. I'd be really interested to like sort the cells from the maternal fetal interface and do functional assays. That's really our dream down the line. But yeah, and your second question was, could we do a different depletion? I would like to. I think we kind of needed to start broader to be like, okay, what are the major players that work? We were wondering if we should do alpha depletion, because to remove both NK and CD8 at the same time, but we didn't go that route. So I think refining the model further, right, to do a more downstream depletion or blockade would be great. So thank you for the suggestion. Thank you all.